In this video lecture, we'll look at cognitive neuroscience methods. We'll investigate 4 methods, starting with lesions, then looking at Penfield's procedure of direct stimulation of the brain. Then we'll look at 2 forms of recording, single-cell recording and electrophysiological recording, and then we'll consider structural and functional imaging. The first method of investigation we'll look at is the use of lesions to study cognitive neuroscience. I'd like to use the example of blind sight from neuropsychology. Here's a short video that shows you this very interesting neuropsychological condition.
Graham is one of the world's few known blind sight patients. He is blind, yet he can see. This paradoxical condition shows just how much our brains get on with running our lives without us even being aware of it.
When I was 8, when I had the accident, it was a road accident that caused the brain damage, I literally used to walk into lampposts. I ran into, these huge great pillars you get in stations. I ran into one of those one day.
The road accident caused colossal damage to the main visual centres of Graham's brain. He can see to his left, but he is blind to everything on the right, in both eyes.
If you put an object in that part of the field and ask him what is it, he has no idea. He cannot perceive it consciously.
Down.
And yet the remarkable thing is, if you move this object, he will tell you which direction it's moving.
Right.
Even though he cannot see the object.
Up.
Blind sight is this term introduced by Larry Weisskranz to describe the ability of people like Graham to detect things but not to be aware of them. So very, very different from what we would normally call vision.
Colin Blakemore is one of several Oxford scientists for whom Graham's mysterious abilities raise baffling questions about consciousness.
I mean, blind sight is extraordinary when you see it. It's shocking. I think it's shocking because it brings home the fact that we can actually manage our brains without consciousness to some extent. And that leads to the question, well then,
why not everything?
Why not everything? And why do we need consciousness for certain things? What is the extra gloss that consciousness gives, if anything, to our actions?
For nearly 20 years, Graham has been the willing subject of a range of experiments that have yielded astonishing results.
You can see things over here.
Oh yes, I can see.
I'm going to move my hand across. You tell me when it appears, when it comes into view.
Now.
Very precisely as it enters the seeing part of your field.
No.
And if I just hold it over here and you look there, you can't see anything?
No.
How about now?
You're moving it up and down.
But you're seeing it.
It's very easy for me to say to you, oh, I saw that move up, Colin. Yeah. And as soon as I say that, you're going to say, ah, he can see. No, I can't.
Okay, let's see if we can just test that a bit more formally. What I'm going to do is make squares that appear on the screen, and they will move either upwards, downwards, or out to the right. You've got to keep looking at this point all the time, so it's in your blind visual field that this is happening, where you're not supposed to be able to see anything at all. But what I want you to do is just to guess what the direction of motion might have been, even though you can't see it. I want you to tell me each time whether you're aware of anything having happened, okay?
Down, not aware. Down, not aware.
Not only does Graham see nothing on the blue side of the screen, he's not even aware of anything happening there at all.
Not sure of that one. I think it moved to the right.
Yet miraculously, he guesses the direction of movement correctly.
If there's one thing that this phenomenon of blind sight teaches us, it is that vision is not entirely seeing, that there can be a disconnection from the capacity to respond to visual information and the actual act of being visually aware of something. Those two things can be separated and probably are in our everyday lives. But the problem is But obviously, we're not aware of the things that we're not aware of. We just don't know the extent to which they play a part.
It's almost as though the patient is using ESP. He can see and yet cannot see. So it's a paradox. It's almost like science fiction. How is this possible? Well, if you look at the anatomy, you can begin to explain this curious syndrome. It turns out from the eyeball to the higher centers in the brain, where you interpret the visual image, there's not just one pathway, there are two separate pathways which subserve different aspects of vision. One of these pathways is the evolutionarily new pathway, the more sophisticated pathway if you like, that goes from the eyeball through the thalamus to the visual cortex of the brain. Now you need the visual cortex for consciously seeing something. The other pathway, which is older evolutionarily and is more prominent in animals like rodents, lower mammals, birds, and reptiles, goes to the brain stem, the stalk on which the brain sits. And from the brainstem gets relayed eventually to the higher centers of the brain. Specifically, the older pathway going through the brainstem is concerned with reflexive behavior, orienting to something important in the visual field, making eye movements, directing your gaze, directing your head towards something important. In these patients, one of these pathways alone is damaged, the visual cortex is damaged. And because that's gone, the patient doesn't see anything consciously. But the other pathway is still intact. And he can use that pathway to guess correctly the direction of movement of an object that he cannot see.
Graham often likens his vision to that of reptiles, whose dependence on unconscious blind sight is powerful evidence for them not having consciousness.
A lizard, if it wants to catch a fly, for example, doesn't actually have to see a fly, it doesn't have to recognise a fly, it just has to be aware of something moving. So if it can detect motion moving to the right, to the left very quickly, it can catch its prey, so to speak, it can eat. And that's very much, I guess, what blind sight is. I'm aware of some of these more primitive functions, but I don't actually see to a right. So I suppose me and the lizard are distant cousins.
One of the goals of neuroscience is to understand which parts of the brain are dedicated to what function, how different mental capacities map onto different pathways and different neural circuits in the brain. And surely this fascinating syndrome is going to help us understand not only the nature of seeing, not only the division of labor between these different pathways, but the question of what is consciousness? What does it mean to be consciously aware of something? Why is one pathway alone conscious, whereas the other pathway behaves like a zombie that's trapped inside him, that's unconscious? The syndrome is so strange that when it was initially reported, people didn't believe it, and there are some people who still don't believe it. But in a sense, it's not that strange if you think about it, because we all suffer from a form of blind sight. In a sense, we experience blind sight all the time in our daily lives. For example, as I am driving this car and having this conversation, all my attention is on the conversation, on the person next to me. And in fact, I'm not conscious of what's going on around me. Even though I'm negotiating all this traffic, avoiding obstacles, avoiding that car on my right, avoiding the car on my left, that's all being done in parallel by another part of my brain, and it never emerges into conscious awareness unless something very strange happens, like a big truck passes by, I might notice it.
Blind sight enables us to steer our way successfully through the world as if on autopilot. Without this zombie in our brains acting on our behalf, the world would be a treacherous place to move around in.
So one method of using lesions to study cognitive neuroscience is to find patients who have brain lesions resulting from disease and trauma, like we saw with Graham in the video. Sometimes lesions occur not because of disease or trauma, but because of incidental surgical destruction. We saw this in a previous video lecture when we looked at split brain patients. For non-human animals, much research has been done looking at deliberate surgical destruction of brain tissue. In the case of blind sight, research has been done on primates where areas of their visual cortex have been destroyed by lesions. A way to simulate a lesion without actually causing any brain damage is to use a process called transcranial magnetic stimulation. Here's a short video that shows how this works.
I'm Roger Highfield. I'm the science editor of the Daily Telegraph. Now, words mean everything to me, but now I'm going to have the speech area of my brain turned off with a giant magnet. Humpty Dumpty sat on a wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again. Humpty Dumpty sat on a wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again.
That may have looked like a strange party trick, but it's actually part of a serious research project conducted by Prof. Vince Walsh in a basement lab at the Institute of Cognitive Neuroscience in London. He's using electrical and magnetic brain stimulation to understand the most complex known object in the universe and how to treat conditions such as migraine and stroke. Just to show the power of the magnetic fields passing through my scalp, and that this wasn't a trick, this is what it did to our videotape, even though it was positioned a few meters away.
Star Trekian. Yeah, very left eyebrow twitch.
It's hard to see because of the interference, but here I could still sing a nursery rhyme, even when he disabled my language centre, because singing depends on the other side of the brain.
Okay. Humpty Dumpty sat on the wall. Humpty Dumpty had a great fall. All the king's horses and all the king's men couldn't put Humpty together again.
I felt a bit apprehensive during this intriguing demonstration, but Professor Walsh thinks nothing of tinkering with his own brain for the sake of science.
Jack and Jill went up the hook. Yeah, that's great.
As researchers from around the world are about to gather to discuss the latest results, he told me about the remarkable potential of brain stimulation.
When we're trying to do experiments in which we establish how people might recover from stroke or some other kind of brain damage, then we can try and make very good, educated guesses about which processes we want to interfere with to help your weakened but still happening processes take over the function that you couldn't do anymore. The equipment and the things we can do with it are evolving into clinical uses now, and we have a number of problems with classical transcranial stimulation with these coils and the big machines and the mapping of the brain that you have to do when you carry out these experiments. What we're trying to think of are ways of getting brain stimulation into the clinic and into the home as well.
So with a migraine, someone feels it coming on, they put these electrodes on the head, and with a bit of luck they can stop the attack before it really develops.
So some migraines are caused by having too much activity in the visual brain area, and some are by having too little activity, and we hope that this can balance out, reverse that relative inactivity in the brain. So we're looking at downsizing the ways of delivering the currents, producing smaller machines, and producing electrodes that will pass very, very small currents that you don't make clicks and that you're not aware of in the way that you were aware of being magnetically stimulated previously. And these will change the excitability of the brain for longer periods of time. The problem with magnetic stimulation is its effects are pretty immediate. You said for a few milliseconds, but then you passed on to the rest of the nursery rhyme. You were fine. With this, we can change the way that the brain is able to be sensitive to tasks for tens of minutes, up to 90 minutes afterwards. And we think that might be much more valuable in a clinical setting.
Jack and...
Boyer performed an experiment that uses transcranial magnetic stimulation to study or to mimic blind sight. He knocked out the primary visual area, we call it V1, using transcranial magnetic stimulation during a simple visual discrimination task. Participants looked at a computer screen and waited for a dot to appear. Their task was simply to identify the color of the dot. After the dot appeared, they were asked if they had seen the dot. If they said they had not seen the dot, then they were asked two questions. First, they were asked to give their best guess whether or not they thought the dot was green or red. And secondly, they were asked to report how confident they were in their previous response. The confidence was recorded on a Likert scale that ranged from 1, meaning they were very unsure of the accuracy of their response, all the way to 9, in which they were claiming to be quite sure that their response was accurate. Part B of this graph shows the results from participants who had received transcranial magnetic stimulation at about the same time that they were viewing the dot in the experiment. For these participants, many of them reported that they were unconscious of the dot, that they had not seen the color of the dot. This is, of course, because their primary visual processing area had been temporarily knocked out. So even though they claim not to see the dot, if we look at the performance of 5 different subjects, we can see that each one of them is more accurate than chance. This dotted line here represents chance performance. So if you were guessing at the color of the dot, you'd only be 50% accurate. But all 5 participants were actually more than 50% accurate. In fact, subject 2 here is almost perfect accuracy, even though they're claiming on these trials to have not seen the dot. If we look at their accuracy as a function of confidence, what we would expect to see is that there's a strong relationship between their accuracy and their confidence. In other words, when they're very unsure of what they saw, then they should also be quite inaccurate. But in fact, again, if we look, 50% accuracy here is chance. If we look at their confidence ratings across the range of very unsure to very sure, we can see that in all cases they're performing well above chance. And the relationship between their confidence and their performance is quite low. This is an interesting experiment because it appears to be creating blind sight in a perfectly normal participant using this interesting transcranial magnetic stimulation technique. Another way to investigate the brain is to directly stimulate it. This was a procedure developed by Canadian neurologist Wilder Penfield. It's sometimes called the Montreal procedure because it was developed at the Montreal Neurological Institute. In this procedure, a patient who is about to undergo brain surgery is woken up temporarily before the surgery, has their brain probed with mild electrical stimulation, and the patient reports what they're experiencing as the various parts of their brain are stimulated. This became a procedure that was quite useful in identifying areas of the brain that might be responsible for focal epilepsy. And in fact, Penfield's work became the subject of a fairly well-known heritage moment video that was produced in the 1990s. Here's a clip of that video. You may have seen it before.
Saul? Saul? Toast is burning.
Toast is not.
Every time she has a seizure, she smells something burning. Now, if we can provoke that smell by probing the surface of the brain, we'll find the source of the seizures. Mrs. Gold, do you feel anything?
I can see the most wonderful lights.
And now what do you feel?
Did you pour cold water on my hand, Dr. Penfield?
Now what?
Oh,
What is it, Mrs. Gold?
burnt toast. Dr. Penfield, I can smell burnt toast. Dr. Wilder Penfield. He cured my seizures and hundreds more. They say he drew the roadmap of the human brain. We just called him the greatest Canadian alive.
A third method of investigation involves recording. We've already looked at this earlier when we were looking at single-cell recordings. Remember Hubel and Wiesel's work on the cortex of cats, where they found different cells that responded to different types of bars at different angles, different sizes, and moving or not moving. We also looked at this in human participants, Remember the Steve Carroll neurons that we discussed in an earlier video? A very popular type of recording is to use electroencephalography, or EEG. An EEG measures the electrical activity on the surface of the cortex using very sensitive electrodes. Now, if we look at these brain waves, what we find is that they don't make a lot of sense. So here's an example where a participant whose brain waves are being measured are presented with a series of stimuli. If we look at what's happening, we can see that there are shifts and movements from negative to positive waves each time the stimulus is presented, but it doesn't look like there's much rhyme nor reason or consistency. However, if you were to take a number of these stimuli, say 50 exposures, and then average them together, what you find is that the averaged waveform, we call it an event-related potential, has a nice, smooth, and consistent shape to it. There are both positive and negative shifts that occur at different times. Most of these shifts are related to some type of brain activity or some type of cognitive process. One of the most popular forms of investigating cognitive neuroscience today is to use imaging. There are two types of imaging. Structural imaging involves trying to get good pictures of brain structures. One of the earliest and most common ways to do this is to use computed tomography or a CT scanner. A CT scanner uses X-rays to take pictures of slices of the brain. Here's an example of a CT slice of the brain. This is a horizontal slice, and you can see this large area in here. Medical professionals often use this technique to try to discover tumors. This is in fact a brain tumor in this patient. If you take enough of these slices, you can fit them together to form a 3D image. So in this figure here, we have all of these multiple horizontal slices that can be put together using a computer program to come up with a three-dimensional model. One of the problems with computed tomography is that it really works best for hard tissues like bones and cartilage. It doesn't work very well with the soft tissues of the brain. Here we're able to pick up a tumor because the tumor is much denser than the surrounding brain tissue. Magnetic resonance imaging is much more useful for taking structural images because it has better soft tissue contrast. Here's a picture generated by a magnetic resonance imaging machine. You can see that there's very nice resolution. We can see all of these sulci and indents in the brain. We can see a clear distinction between the two hemispheres. It gives us a much better picture of the soft tissue in the brain. And just like CT, you can also take slices. These are vertical slices through a brain, and neuroscientists can use this to search for tumors in the same way they might use a CT scan. A second type of imaging involves functional imaging. Rather than focusing on the structure of the brain, functional imaging attempts to measure the activity of the brain. The earliest method involved using positron emission tomography or PET scanners. A tracer is injected into the participant that has a mild radioactivity to it. The tomographer reads the location of these radionuclide, and by doing so is able to follow the flow of blood through the brain. It turns out your brain is quite an oxygen consumer. It consumes a lot of oxygen, and the way to get oxygen to the brain is through the blood system. So when some part of your brain has to do some processing, we need to get oxygen to that part of the brain, and if we follow the blood flow, we can follow the parts of the brain that are most active. This is what we mean by functional imaging. We're measuring what the brain is doing. You can combine this then with a CT scanner and have location information about the precise parts of the brain that are involved in the processing. Here's an example. On the left column, we have original PET images of the regional cerebral blood flow of a participant who is at rest. These are the top two images. At the bottom, we have the same brain activity measured during a language activation task. The task was to just silently generate verbs from auditorily presented nouns. So if the researcher said bat, the participant might say hit. If the researcher said ball, then the participant might say kick. The right column shows the cerebral anatomy for the particular slice we're looking at, and at the bottom, the same image showing activated areas. The way these activated areas are determined is simply by subtracting the brain activity at rest from the brain activity during the task. And when we do that, we can see that there are areas lit up here. This view of this horizontal slice of the brain is from the top, which means that this is the left hemisphere. We've talked about cerebral dominance before. This is yet another example of how the left hemisphere is dominant for processing of language, in this case, this verb generation task. Today, most imaging is done using MRI, but rather than look at the structural aspects of the brain, the MRI machine is programmed to detect oxygenated blood. So just like the PET scanner, it can follow the blood flow around the brain. So here's an fMRI image of a brain, and what's typically done is areas of activity are given these nice color shading, so we can determine which parts of the brain are very active and which parts of the brain are less or even inactive during some kind of cognitive processing. Functional magnetic resonance imaging has become hugely popular in cognitive neuroscience. However, there are many problems with functional imaging that are rarely discussed. When we think about what's happening here, this is very similar to the phrenology that we looked at earlier, except the phrenological approach that's being taken is much more technologically advanced. Nevertheless, it's depending on this assumption of localization of function that we talked about in one of our previous videos. One of the problems associated with magnetic resonance imaging and PET imaging as well is that it relies on this method of subtraction. Remember how we tried to determine what parts of the brain were active by subtracting the activity at rest from the activity while the participant was doing the verb production task. We saw subtractive methods before when we were talking about reaction time in the first module. I mentioned then that contemporary cognitive psychologists rarely use a subtractive method on reaction time. It's not considered to be a strong experimental technique. Most researchers involved in mental chronometry use other methods. Nevertheless, imaging relies very heavily on the subtractive method, and it's riddled with a number of problems and assumptions that may or may not be met. Another issue concerns localized versus distributed processing. Communication between different regions of the brain is important, and measuring activity in localized areas doesn't give us information about how these local areas are communicating with other local areas. A lot of the cognitive processing that we do involves multiple brain areas in communication with other areas so that they can work in concert. Another problem with functional imaging is that it relies on an indirect measure. We're not actually watching the brain in action. Remember, both PET scanners and functional magnetic resonance imaging follows blood flow. Oxygenated blood going to different parts of the brain is thought to indicate that that part of the brain is active. But getting oxygen to the brain happens after the fact. After I've used part of my brain, it's starved for oxygen, so my blood brings more oxygen to that part of the brain. So you're not actually measuring activity as it happens. You're measuring activity that happened in the past. We know it was active because blood is going there. But there's a delay between when the blood gets to a part of the brain and when the activity occurred. In the case of PET scanners, that delay can be up to 60 seconds later. In most fMRI studies, the delay between blood getting to part of the brain and the time at which the processing occurred is usually between 2 and 6 seconds, although sometimes it can be 15 or 20 seconds later. So we're not directly measuring activity of the brain, we're measuring blood flow after the fact, in fact, considerable time after the processing occurred. In fact, after a considerable amount of time has passed since the processing had occurred. Another problem is that even though the pictures look very nice, they seem to be nice, sharp, structural pictures of the brain and lots of pretty colors added to it that make it look very impressive, the resolution is not as good as we might like it to be. So for example, in a PET scanner, the resolution is approximately chunks of cortex between 6 and 9 cubic millimeters. fMRI is a little bit better. It's about 2 by 4 millimeters. That would be about the same size as a grain of rice. We call this area a voxel. This is the volume of brain that we're measuring, and it's like a pixel. Pixels are the dots that make up your computer screen. When you put volume and pixel together, you get the word voxel. So these areas of investigation, these small grain-sized areas of the brain that we're measuring, we call a voxel. Now while that may seem like a pretty small piece of brain, neurons are small. One voxel can contain millions of neurons. And of those millions of neurons, you would have to have a lot of them light up and be active for the fMRI to be able to pick it up. What if within that set of millions of neurons there are smaller cell assemblies of neurons that might be quite active, but because you're measuring across millions of neurons, it would be very easy to miss the activity of these smaller cell assemblies. Another problem we have is individual differences in brain organization. And this is really a key problem with the whole notion of localization of function. So for example, we talked about Broca's area. And Broca's area is found just above the temporal lobe towards the front of the brain. And we know that it's responsible for processing, for producing speech. But not everyone's Broca's area is in the exact same place. It can vary from person to person. Here's an example of brain scans taken from 6 different participants who were taking part in a spatial memory task. These are horizontal slices, and the colored areas represent areas of activation. If we just do a comparison between these 6 participants, we can see that there are some commonalities, but no 2 of these scans look the same. If we look at this person over here, for example, we can see lots of activity happening in this part of the brain, but it's not happening with these other participants. And this participant has lots of activity in this part of the brain. This participant has none. There's a lot of what we call individual differences in these activations. And it's not as simple as a phrenological map where everybody's processing is in precisely the exact same place in their brain. There's actually quite a bit of variability. Another issue that arises with imaging are statistical issues. because we're looking at blood flow, usually what we end up doing is summing the activity in a voxel over a period of time, maybe 5 seconds. How much blood is flowing to that area in the 5 to 10 seconds following processing. And then what we do is sum the total amount of processing that's happened over that period of time. Well, the problem with doing this is that something interesting may be happening over those 10 seconds. Is the amount of blood flow constantly increasing? Are there fluctuations in the amount of blood flow? Does it rise, fall, and rise again? We don't know because we sum over time. A better approach might be to look at how the blood is flowing across time to see if there are changes across time. This, of course, would be quite complex, although there are some researchers who are doing it. Another statistical problem is the fact that we are doing an analysis on hundreds or thousands of voxels. So if we break the cortex down into rice grain size parts, there's lots of different voxels that we're going to be looking at. Now what we want to know is, of course, there's random fluctuations in brain activity that we can't really account for. We want to make sure that the activity that we're picking up is actually activity due to the processing that's being performed. Now to do that, we tend to use a statistical rule where if the probability of something happening by chance is less than 0.05, we assume that it didn't happen by chance, but it happened because of an experimental manipulation. So if the blood flow to a particular area is, the amount of blood flowing to a particular area is greater than what we normally expect, and in fact we wouldn't expect that much blood to flow randomly for a random reason less than 5% of the time, then we would say that we're not sure that this is really due to our manipulation. We call it the 0.05 rule, but it's basically a way of trying to avoid situations in which changes that we're observing are due to chance rather than due to experimental manipulation. Well, the problem is this 0.05 rule can be applied to hundreds of different voxels. This means that we could end up with a number of these voxels appearing to be increasing in their activity, not because of what we're processing, but because of chance. Think of it this way. Imagine you have a 20-sided dice. If you roll a 1, that's quite rare. It should only happen 5% of the time. But if you get to roll the dice 1,000 times, you're going to have lots of 1s appear. So even though the probability of rolling a 1 is only 5%, because you're rolling 1,000 times, we're going to expect to see that 1 appear multiple times. And it's essentially what's happening in an analysis of several voxels. If we're looking for activity that's increased more than we expect by chance, but the dice we're rolling is a 20-sided dice, it means we're going to have multiple voxels becoming active. Researchers who do this kind of work try to be careful to correct for all of these comparisons that they're making and reassign this rule. But there's a clever experiment that was done by Bennett that I think exemplifies this problem quite nicely. In their study, participants were asked to determine what emotion was being expressed by an individual in a picture that they were looking at. So while in an MRI, a participant would get a series of pictures, and these pictures depicted individuals in social situations with very specified emotional valence, and then the participant simply decides what emotion is being expressed. Kind of like the man that I keep asking you about if he's angry. This uses a subtractive method where a comparison is made between the brain at rest and a brain making these social emotional expressions. These were the results they published. If you look closely, you can see these red areas are associated with significant changes in activity. This is pretty typical of a fMRI study. We see these nice MRI pictures with areas of activation that we think are related to the processing, in this case, the processing of emotional stimuli. Now, if you're looking at these pictures and thinking that they don't look much like a human brain, that's because the participant in this study was a dead salmon. You see, these areas have lit up. Bennett was actually simply calibrating his experiment when he collected this data. So they wanted to put some live tissue into the MRI machine to make sure the experiment would run properly. These experiments are very expensive to run, so you do want to make sure it runs properly. So to do that, they had live tissue, a dead salmon. They put it in the machine. They even presented it with the pictures, just like they would a real participant, and then did their analysis. But because they didn't correct for those multiple rolls of the dice, we find these areas that become active. I think it's an important warning to people who do MRI research. That's it for Module 2. In our next module, we'll look at perception.